the speaker, but actually Professor Jin-Soo Kim at Postech has worked with Alexander Hoffman for last couple of years, so he knew way more than me for Alex. So I gave him a favor that introduce today's speaker. So Jin-Soo, please.
Thank you. Yeah, it is a really great pleasure to have Alex at IBS BioMed Korea. So it is also very great honor as one of the collaborator of the Alex to introduce him. Alex is the founding director of the Institute for Quantitative and Computational Bioscience at UCLA. He is also the Thomas Usher Professor in the Department of Microbiology, Immunology, and Molecular Genetics at UCLA. He obtained his PhD from the Rockefeller University in 1994. He also worked at MIT as a postdoc. Before joining UCLA, he was professor of biochemistry at UC San Diego. There he founded the San Diego Center for Systems Biology. Interestingly, he holds many degrees in physics, geology, biochemistry, and molecular biology. His research interest is the development of a predictive understanding of immune responses. His lab aims to understand how signal systems such as NFkB signals control and reprogram immune systems. His lab has published lots of fascinating works to many prestigious journals. And definitely I cannot introduce all of them for this couple of minutes. So I'm gonna pass my mic to Alex. Welcome.
Oh, thank you so much for this kind introduction, and thank you so much for this invitation to come to you from UCLA to Korea and your really exciting institute in mathematical biology. It's going to be really exciting to see how you grow the community and how you grow your local community and how you reach out to all of us here. It's one of the benefits of this pandemic is that we're able to come together so easily, and I really appreciate the opportunity to talk to you. So I will just begin to share my screen, and I think I'll work. So and please interrupt me when things are not clear, or there's an urgent question. That's absolutely fine. Otherwise, there'll be time for questions later on. And of course, my talk today, as you heard, I think my background is largely on the experimental side, but we do aim to develop mathematical models. And so collaborators who have a deeper understanding of the dynamical systems that we study are really, have always played a really important role in our research. So I'll talk to you about a temporal signaling code that allows immune sentinel cells to determine their responses to infection. And so what are these immune sentinel cells? Well, they're cells in the body, in every tissue and every organ that are the first responders to pathogen. And macrophages, tissue-resident macrophages are a good example of that. And they must respond to any invading pathogen, whether virus, bacteria, eukaryotes, and also detect tissue damage when cells are injured either through heat or through lysis or anything else. And they're capable of a variety of different responses. First, they have the capacity to defend against these pathogens, actually by engulfing the pathogens, and by, or if they get infected, by committing cell death and taking the pathogen with them. They also are able to recruit other immune cells to the site of infection. and thereby try to localize the infection and resolve it. And the number of different mechanisms by which they can do that. And then finally, they're also able to activate the immune system systemically, and that leads to adaptive immunity, including T and B cell activation. So macrophages can do all of these different things. And so because this is so important, it's been studied by many, many people. And we know many of the molecular players inside a macrophage that are involved in the detection or the signal transduction, or in fact, the effector proteins. And so when I show you this, first, before I show you the map, it's when these processes are not working well, many different types of diseases are the result, autoimmune diseases or immune deficiencies and so forth. So these systems have to work well. So many people have studied this. And so this is the map of the molecular network that's already, this is the state of it almost 20 years ago. Kitano and co-workers put this together. And here you see some cell membrane receptors that are there to detect pathogen. And here is a nucleus with transcription factors. And in between is a molecular network. And so, I show this slide for once to tell you that a lot is known, but at the same time, this map actually does not really tell us how things work. Because if you follow the lines from each of these receptors, you will probably find all the way down that everything down here is activated, which means that there's absolutely no specificity in this. So that means whether it's fungi or viruses or bacteria would activate exactly the same response. So there's a lot of information that is missing on this map. There's quantitative information missing. There is timescale information missing, and there's abundance information missing, and so forth. And so this map really does not yet answer the question about how do you generate specific immune responses. One thing that is remarkable about the network is that it's highly dynamic. So in response to a cytokine or pathogen, you see a transcription factor NFkB being activated repeatedly in an oscillatory pattern. And I think some of you have seen these before, and Mike White's group showed in single cell studies also that there is surprising oscillations. So this observation led to the hypothesis that actually the dynamics of signaling represent some kind of code that allow the cell to produce different types of responses, so that a single pathway is activated not just by a on-off switch, but by transducers that produce temporal patterns, sort of like the old telephone, analog telephone, that produce NFkB responses of different temporal patterns that then might lead to different types of cellular responses. So this hypothesis is now more than 15 years old, and yet it was actually tricky to figure out whether this is really true. So one question is, if we're saying that there's a code or a language, the first thing is to actually identify what are the words in this language? What are the code words? Or what are the packets of information that are informative about the pathogen or the outside environment that can be the code word or the temporal feature that informs the nucleus about the outside. So what are the code words? And so this is the question that we tried to address. And we thought maybe instead of taking a hypothesis approach here, what we really need to do is just listen to cells and generate a large amount of data that we can subject to some kind of statistical test. And so Brooks-Taylor produced a mouse in which NFkB is fused to this fluorescent protein, and he developed a experimental and then automated image analysis pipeline that allows him to see whether NFkB is in the nucleus or whether it's in the cytoplasm, and then quantitate this and produce the trajectories over time. And this is now published, and the work was continued by Ade Adelaya after Brooks left. So this type of system can give us these types of movies where we see in response to TNF, a cytokine, we see here that NFkB is in the nucleus, or it goes out of the nucleus, and these are in real time over several hours. Actually, not in real time. I'm sorry, this is of course time lapse here. And the pattern is oscillatory, but the same cells, when stimulated with LPS, a different ligand, you can see that NFkB remains in the nucleus for many hours in a largely non-oscillatory manner. So the cells are capable of very different types of responses. So you can plot many cells here. These are about 500 cells plotted over time, over 12 hours here. And you see the heat, the red indicates nuclear localization and the blue is cytoplasmic localization. You can see that every cell sort of oscillates, even at late times, but they're asynchronous, and that's why the average doesn't really show what's going on. So this is a highly oscillatory pattern. But in response to LPS, the response is largely non-oscillatory. And so, but I think rather than focus just on these two conditions, we decided to generate a lot of data in different types of different stimuli doses and different types of ligands, TNF here, pAMP-CSK, and CPG and LPS and poly-IC. And so in each case, we have hundreds of cells over many hours, so millions of data points that we can then begin to analyze. So oscillations may be one interesting feature, but there are probably many others. And so we decided to generate a large library of whatever we could come up with, 918 features. For example, the amplitude at a particular time point, which is what we call time series data. Or each time point can be characterized by the total activity to that point, the total integral, or the derivative at that particular time point, the slope, whether it's positive, negative. That's how the data could be described. Or particular features like the peak amplitude or the peak time or the duration over a particular threshold. Or we can, do Fourier transform and identify the peak frequencies or secondary frequencies and see whether they are informative about something. So with this library of features, we thought, what we want to do is really correlate these features to the known stimulus, the dose and the type of stimulus. And so for that, we used an information theoretic approach, Claude Shannon's approach to measure mutual information or channel capacity, where the stimulus, whether it's a particular concentration of a cytokine or pathogen ligand, is the source of information, and the destination is, in this case, NFkB dynamics that are being associated with the nuclear target genes. And so and it's a correlation test, if you will, and so we are able to identify the amount of NFkB or each of these features of NFkB dynamics with the stimulus. And we're asking how do we maximize the amount of information by using these different features.
Mm.
So you can see here that if we use just one feature, we can get up to almost 1 bit of information. But if we use the top 2 features or the top 3 features, always used combinatorially, selecting the top 3 or the top 4, then we are able to capture more of the information in the data set. Until the metric plateaus out at around 2.1 features, 2.1 bits of information, with about 7 features. 7 of these features provide almost all of the information.
So what is the meaning of 918 features? So you have all these different type of characters, like a time series, integrate activity, and that kind of series. You have about 900.
Yes, yeah. So there we enumerated, 918 features and it's an ad hoc approach, if you will. And there may be others that you could come up with, but it was a large number, and so we're selecting from this large number.
I see. Wow.
And so of those 918, 7 provide non-redundant information, and if we add more and more features to our algorithm, they are not providing any more information.
I see.
So the question is, how many, what are those 7? Those are the informative features.
Right. Rank.
And so 2 of them are derivatives at the early time. They basically characterize what the speed of activation is. And then there is a peak amplitude that at a certain time point that determines the peak. There's another derivative that is typically negative, that determines whether the activity has an oscillatory component, whether there's a rapid downregulation. There's also total integral of that is one of the features. There is the time to half maximal integral. So that's kind of like how much activity is early versus how much is late. And then the duration over a pretty low threshold. So the total duration of activity also provides non-redundant information, although we already have, of course, total and peak and so forth. So essentially, there are 6, if you will, dynamical features that convey information to genes about the outside of the cell. And because they're informative features, there are, if we use the analogy that this is a code, then these would be signaling code words, which then the immunity editor decided that this was a nice term, signaling codons, analogous to the fact that we have genetic codons, which are packets of information that are being transmitted from generation to generation. And here, these are packets of information that are transmitted from the outside of the cell to the nucleus.
I see. So, so Alex, there was a question from audience. These features extracted from population average data or single cell measurement, I guess?
Single cell measurements.
Yeah, single cell measurement, right?
Single cell measurements, these types of measurements, this entire library of measurements, basically. Yeah. And so it's basically a big data approach of identifying informative features. So you can see that these features, when we then measure them, are deployed stimulus specifically. So in response to TNF, the speed feature is high. It's slow, it's lower. This is a histogram of, violin plot of 500 or so cells, With CpG, it tends to be lower, and with poly-IC, even lower. And total activity varies also in a stimulus-specific manner. Peak amplitude doesn't change a whole lot because here we're looking just at the highest doses, but lower doses, the peak amplitude is distinct. The oscillatory feature is particularly high with TNF, but not very high with any of the others. And then duration, it varies and so forth. So each of these signaling codons are deployed in a stimulus-specific manner. So this is a cytokine, this is a bacterial ligand, a different bacterial ligand, a third bacterial ligand, and this is a viral ligand. So virus, bacteria, and cytokine. And so here's another way to sort of a graphical abstract way. Here are the different receptors, and each one produces these particular features in a stimulus-specific manner.
Gotcha. I see.
So if these are informative features or code words, then we thought, and they constitute the code words of a language, then we should be able to teach that language to somebody who's otherwise not particularly smart, which is our computer. So using a machine learning approach, like an ensemble of decision tree algorithm, we can take all 918 features, or we can take the 6 that we identified. We can generate a model, and then we can ask the model how well it recognizes the true stimulus when it's presented just with the dynamics of NFkB. Right? So this is the recall here. The precision of recall is the F1 score, and we see that it does reasonably well for each of these stimuli. And you can see particularly that the 6 codons do almost as well, or pretty much the same as all 918. So that means the 6 are informative, and they're also sufficient, right? And then further work showed that if we take one away, then we lose performance.
So here, what did you predict?
We present, so after training the model, we presented the model with NFkB trajectories, and then the model had to predict what the ligand was.
I see, gotcha. Thank you.
So it's the model, if you will, is in here is like what you would want the nuclear genes to do. Right? The nuclear genes only see the dynamics of NFkB, and then they need to know whether the cell has to fight a bacterium or they have to or a virus, right?
Got it.
So of course, machine learning classifiers only allow you to look at the precision, but you also can see very nicely where there's confusion.
Oh,
right? So in this case, what we see there is some confusion here, particularly PAM3CSK with LPS. But luckily, both of these are bacterial ligands. So actually, it's a similar kind of pathogen. And so the fact that there is that the specificity is not high might be okay. So if we redo the classifier and we just take all the bacterial ligands together, then actually we see much better performance. So then
Alex,
we ask,
may I interrupt you?
Absolutely.
So I think you used all the about 900 features to train the machine learning algorithm. What if you use only the 7 or 6 you specified in the beginning?
That's
right.
You have a better, similar accuracy as the 900?
Yeah, that's what is indicated here. These are the 900, and these are just the 6.
6 codons.
Oh, I see. I got it. I see.
Do you see my cursor?
Yeah.
Cool. Okay. So yeah, so then I think from then on, because actually using 918 in this model takes it computationally takes a long time. So all the work, subsequent work is with the 6.
So this indicates the codons you found is also, I think the codons you found is biologically searched features. by using the Shannon capacity, am I correct?
Yeah, so we use Shannon capacity to identify them as informative, and then we use machine learning to show that they're
Wow.
sufficient.
I see, interesting. Thank you.
So because it's like a sufficiency test, right? The Shannon capacity doesn't tell us that we really have everything, but this is more like it. So
got it.
Okay, so so then we asked, is this biologically of interest? So we have this reporter mouse with GFP in it, right? And so we bred it into a mouse model for this human inflammatory disease, which shows up sporadically. Every now and then, people develop symptoms that are very unpleasant. It's an autoimmune disease. But typically, the symptoms only show up sporadically, and in the same in the mouse. And so we isolated macrophages from this mouse, and we measured NFkB dynamics, and then we looked at the codon deployment. So here, and we used, in this case, just 3 stimuli, TNF, LPS, and poly-IC. You see that the speed is much lower with poly-IC in the normal mouse and also in the diseased mouse. So it's pretty normal. Peak amplitude also looks pretty normal.
I see.
But oscillatory, non-oscillatory is very specific here, but is not very specific at all. It's just always non-oscillatory. And there's no distinction. So we've lost the stimulus specificity in one particular codon. Total activity is pretty much the same. Duration has also lost the stimulus specificity. And early versus late is also pretty similar to healthy. So it's interesting from 2 points of view. One is that there is a defect in signaling, but also that the defect can be localized to 2 features. And so what is the outcome of that? Well, if we train another machine learning algorithm, a machine learning model, we see good precision in the healthy macrophage between these 3 ligands. And in Sjögren's, we see a big problem in a couple of these, particularly when the true ligand is TNF, it's often misinterpreted to be LPS. So what does that mean? That means if a neighboring cell signals to the macrophage, the macrophage may misinterpret that there is a bacterium present, that a pathogen is present, even though the signal came from a neighboring cell.
I see.
And so that might lead to, an exaggerated wrong response, and that may lead to the symptoms. So just to conclude this sort of first part here, I think what we did is we used developed sort of this high throughput experimental image analysis workflow that allows us to listen to immune cells and generate a lot of data. And then we analyze this data using an information theoretic and machine learning approach to identify code words of this language. And they're called signaling codons. And then we find that in at least one immune disease, autoimmune disease, it appears to be characterized by a level of miscommunication due to misrepresentation of 2 of these codons. And so that means these types of diseases might be called signal confusion diseases because there's increased confusion. With that, we now know what those codons are. We need to want to know how they're actually encoded, right? And so what does the molecular network do to create these dynamics? And the basic idea of NFkB signaling is that there's a negative feedback loop,
Right.
and that negative feedback loop is driven by IKB alpha, and it is the signaling is activated by a kinase whose activity is controlled by different receptors. And each receptor, of course, has adapter proteins and so forth. So this negative feedback loop is known to generate oscillations, and that's great. But we also saw not only oscillations, we also saw no oscillations.
Mm-hmm.
So the oscillations that we saw in response to TNF are in fact dependent on IKB alpha, just like we expected. If you knock out IKB alpha, then you see no oscillations. But the question is, how can that same normal network that provides these oscillations, how can it also give a non-oscillatory activity, which we saw? So we did a Hopf bifurcation analysis of our network.
Mm-hmm.
And so here are, we're plotting the steady state as a function of how much IKK there is, how much IKK signal there is. And we see a bifurcation event at some low level of IKK that indicates an oscillatory regime. And we see this regime, this phase 2, we see oscillations of NFkB. And then regime 3, so we see that this bubble of this actually begins to close. And we see that the oscillations begin to dampen. And then at even higher levels of IKK, the bubble has closed and there is a single steady state, and indicating a lack of oscillations in this fourth regime. So, and interestingly, the level of IKK, while it controls whether you have oscillations or you don't have oscillations, it does not control the frequency very much. Mm-hmm. Frequency remains stable. So, or the oscillatory period. So it always is between 1.5 and 2 hours, basically. Mm-hmm. And so this is actually interesting that you have this negative feedback loop whose oscillatory behavior is dependent on the amplitude of the input. Mm-hmm. And we can talk more about that if there are questions about this, because mechanistically it actually makes a lot of sense. So that means all that is needed to generate oscillations or not oscillations is to control the amount of IKK, the magnitude of IKK at different times. And that's what these guys do. So each receptor with associated proteins will activate IKK at different times. And so if I provide more detail, then here's the core modules of our math model, our ordinary differential equations model. Uh-huh. And here's IKK, and it receives inputs from each of these receptors. Either the receptor in the plasma membrane activates a few molecules, ultimately IKK, or in the case here, it's plasma membrane and endosome inside the cell. There are a couple of inputs that converge each of these things. So notice the models are very simple. There is a stimulus, it has a half-life. There's an activation of the receptor. And then there is an adapter protein. And this adapter protein has a high Hill coefficient and is very nonlinear, whereas this adapter protein is quite linear in its response. And then there's an activation of IKK. And that's basically all there is to these models. And so the models are very simple, and yet, as I can show you here, we basically can recapitulate the behavior of oscillatory NFkB in response to TNF. non-oscillatory NFkB in response to TLR4. Here's the data, here's the simulations. Or in response to TLR9, or in response to, oh, sorry, that was TLR3. That should be 3, 9, and TLR2, and so forth. So, and then, so to some extent, the model works. Now, if we, and so that part was published, but what we're currently doing, and we haven't completed that yet, is to actually try to recapitulate here the single cell data sets and the distribution of responses in experiments that you see here, the diversity of responses, by distributing parameters. Here's a distribution of parameters, and by distributing them, we can try to recapitulate also the distribution of responses. 1 nanogram of LPS model simulation matches pretty well the experiments, LPS, and so forth. And here with TNF, which is oscillatory, here you see that this type of model doesn't quite do that well yet, right? The oscillatory behavior is longer lasting than what we see in our model. So we still have some work to do to really refine this. But this is the direction we'd like to go in, to then establish a virtual cell, if you will. So just to conclude this part is that these, we have an ODE model of the molecular network that recapitulates the observed signaling dynamics. And the signaling codons are encoded by a common core module and a receptor proximal modules that feed into the common core. And the common core is responsible for the oscillatory codon. And the receptor proximal modules are responsible for all the other codons. And they're controlled by the ligand half-life, receptor turnover, and Hill kinetics in the adapters.
I see.
Basically, those are the three things, and how they interact with each other, that's what determines the deployment of each signaling codon in each condition.
So, Alex, in your core module, I guess the NFkB make a repression for IK beta via, I guess, mainly sequestration, I guess.
Yes. Yeah, sequestration. That's right. So let's take a look at this. It's a so it sequesters NFkB with IKB.
And then what happens in the nucleus?
It also in the nucleus, right? So so let's see. Let's take a look at IKB. IKB is here's IKB gene. It's the mRNA is made, the protein is made, and as a free protein, it is capable of decay, or can go in the nucleus, bind NFkB, make a complex. The complex tends to get exported, and when it's exported, it is subject to IKK mediated decay. So that's the and then you have no NFkB in the nucleus, and that means but IKB, it's in the cytoplasm now, and now IKK can degrade this, and then can free NFkB and goes back in the nucleus. So that's how you get the oscillations. And
NFkB also can bind to the some gene so that it can repress it, because there is some that there.
Yeah, only the IKB alpha gene in this case. Yeah,
I see.
And but now you can see that when IKK is very high, what happens is that the IKB is made, the free IKB, and before it has a chance to go back into the nucleus, it can be degraded by IKK.
Oh, I see.
And that's how high IKK you get non-oscillatory activity.
I see. So do you know any study which disrupt the binding between NFkB and IKB?
With a drug?
Yeah, whatever.
I don't think there's a drug that disrupts the interaction, actually.
I see. There is even no mutation about that kind. This is the question.
Well, people have tried to make mutations, but the problem is it's a very large surface, actually. So you can make mutations or truncations, and then but then you don't really have a viable protein.
I see. I see. Gotcha. Okay. Yeah. Thank you, Alex.
All right. Let me get to the last part, which is after I told you how these signaling codons might be encoded. Now I want to tell you how they might be decoded.
Oh, so sorry. There is another question from audience. Shin Dong-gun, he asked, can this signaling code analysis be used to find a novel biomarker for a certain disease? If so, do you have biomarker discovery case studies?
That's a great question. You know, the difficulty, of course, is to, I mean, in principle, yes, I think the dynamics of NFkB signaling is very much indicative of the health of a cell, very much so. But the problem, of course, in humans is that we don't have GFP in our cells and we can't do live cell imaging. So we need to, so I think determining the dynamics from human samples will be close to impossible. So we will need to find some other markers of the dynamics.
Right.
And so we're actually trying to do that by looking at genes that are indicators of particular codons. And then maybe those are good candidates for biomarkers.
I see.
But yeah.
Thank you.
So yeah, so I told you about these codons, and actually we've been working on trying to understand how genes decode the codons. And so there was a paper by Suzanne Godet's lab that looked at how amplitude is decoded through an incoherent feed-forward loop. We published a study on how duration is decoded, also using math modeling and data sets at a chromatin level. There's a slow chromatin opening step that requires a certain amount of time. That is the key for genes to determine the duration responsiveness. And then we now have a paper in press with Mariko Okada on speed. But of course, the most interesting thing that we all love are oscillations and the lack of oscillations. How do genes decode the presence or absence of oscillations? And that is what was recently published in Science, and I'll just tell you briefly about. So the backdrop is that some time ago, we did an experiment to address this, and we used oscillatory cells or non-oscillatory cells, and we looked at some prominent immune response genes that are activated.
Right.
But in either case, we see the same level of activation. So at that point, back in 2005, we were quite frustrated.
Right.
I mean, we were happy that Science was interested in publishing our study, but we were a little bit frustrated because this is a really interesting feature and we saw no difference.
Right.
So then we thought more recently whether maybe we're looking at the wrong things. We only looked at immune response genes. Maybe instead of looking at immune response genes, we should look at the chromatin, the chromosomes more generally. Remember, of course, that chromosomes, most of the chromatin is packaged into nucleosomes that are tightly packed, and the genes are actually not available. They are closed, they're packaged. And only some genes, the immune response genes that are poised, they are open. But these places there might be some genes here, but they're not available for activation. They're closed. So we're wondering whether maybe NFkB has a role in opening them. And so and so here is a structure of this nucleosome. And you see that DNA wraps around the nucleosome about almost 2 times, 2 turns, 1 3/4 turns. And the DNA is on the outside. And oops, sorry about that. And when we have a structure of NFkB, here's the DNA, and we're just looking at the axis here. And you see that NFkB actually completely wraps its arms around it. Mm-hmm. There's no way that NFkB can actually bind to DNA that's on a nucleosome. Mm. So the two are mutually exclusive. So if NFkB wants to bind this, it first has to wait for some molecular stochastic breathing of this, where this is slightly unwrapped, and then NFkB can probably bind there. And then when it binds there, it has a chance to continue the unwrapping process. So we developed a model for this, and this is Catherine Shu's work, and now Jinsu has been really been involved in this, where actually there are 14 contacts here. And so if all 14 contacts are, then the nucleosome is in a fully closed position. And when all 14 contacts are lost, then the DNA is completely open. And so this is the open DNA, and the 14 contacts completely closed, and they can open sequentially. And the sequential opening can be mediated by NFkB through a Hill function. So that's this very simple model. Of course, there may be many different pathways, and Jinsu is the expert at this, but in this first version that I'll tell you about, we made the assumption that this is a very simple linear process. And so with this model, as you probably intuitively can imagine, if you pick the right parameters, you will find that oscillatory inputs, here we show oscillatory inputs, will lead to only partial opening, but never complete opening. Right. But if the same amount of activity, when it's non-oscillatory, can lead to complete opening. Right. And the same is true, if you vary, for example, the amplitude, different amplitudes, you need a certain amount of amplitude to get non-oscillatory thing to open. But even if you add more amplitude, oscillatory activities will never open this thing completely. Right. Or the total activity, you need a certain amount of total activity, but if you add more total activity, then the oscillatory guy won't open it. So, so that's that's the basic sort of theoretical behavior. But then we thought, let's take the measured activity of NFkB. These are the NFkB dynamics that we've measured in the first part of the talk in response to TNF, oscillatory, bacterial ligands, non-oscillatory. And now we used each of these trajectories as an input to the little model I showed you. And we're showing how open the chromatin is. And we see that these activities don't open the chromatin, but these activities will open the chromatin. And so what's interesting about that is we can measure chromatin opening using a next-gen sequencing approach. And we find that there are about 4,000 locations in the genome that show some opening. 2,000 locations that are opened actually maybe have nothing to do with what we're interested in, because they have a binding site in these locations that is not NFkB binding. But there's another 2,000 locations that do contain an NFkB binding site. And they are opened with LPS, with CpG, and PAMPhi-CSK, but not with TNF and not with poly-IC. just like our prediction. This is TNF, this is poly-IC, and these guys can be open just like the measurement. So actually, this simple model made a prediction that we could measure and therefore confirm. So, and then we did another experiment where we knocked out this negative feedback as I did before, and we used these oscillating cells, non-oscillating cells. And again, the non-oscillating cells open the chromatin much more than the oscillating cells. And then finally, is that these open chromatin regions, those same regions that are open, so we're generating open chromatin by non-oscillatory NFkB, and then we wait about a day, and then we stimulate the cell with a secondary stimulus. And we find that in the control, the secondary stimulus does not activate gene expression, but in the cells that had chromatin opening with non-oscillatory NFkB, you then get gene expression. So these open places can now activate new genes. And those new genes are immune response genes. They play a role in immunity. And so this is exciting because, you know, we know that we can train our immune system to be more powerful. And maybe this is one way that NFkB can do this, by using non-oscillatory NFkB. So just to wrap up, I think what I tried to show you that there is, in fact, this idea of a temporal code exists. It's not just a hypothesis. And I've tried to address what the code words are of this code. I try to show you how they might be encoded by the molecular network, and how they're decoded by genes or by chromatin. And of course, here we used mostly statistical machine learning approaches, and here we used math modeling ODE approaches. And the decoding part on chromatin, of course, these are single molecule events. And so stochastic simulations are probably a more accurate way of representing that. And that's what Jinsu has done and will probably tell you about more in the future. So with that, I just want to thank you for your attention, and also my group who've contributed to this work. Thank you for your attention.
Wow, thank you. Wow, great talk. So yeah, please, if you have any question, just you can unmute, and please feel free to ask a question to speaker.
There might be a question in the chat room.
Oh yeah, chat room from Bongun Shin from he's asked, for an animal experiment, how much did it approximately cost to figure out all the dynamics you presented in the Sjögren syndrome case? How much? Wow, money.
You know, I think actually the experimental pipeline is pretty much automated. So of course, setting it up took 2 or 3 years. But now that it's up and running, you can, this particular mouse model is available at Jackson Labs, you know, and so we can buy the animal and then for $100, and then we do need to breed it into the reporter mouse, the GFP reporter mouse. So that will take 4 months or 6 months or so. So that it does take that long, just the breeding. And then you do the experiment, and then after getting the experiment, you know, the experiment takes a day, and then analysis another day. It's pretty automated. So it's not a huge amount of hands-on work, but we need the reporter. We need the fluorescent reporter.
Right. Okay, cool. Any question from audiences?
Can
Yeah,
I ask something? I think in your second part, I think you mentioned about distributing parameters to recapitulate single cell heterogeneity in the system. I think I was lost at that part. So could you explain how did you distribute the parameters a little bit more?
Yeah, great question. And that work is certainly not done. So we have published a paper a few years ago in which we distributed only 3 or 4 parameters in the system and tried to show which ones are more likely to be responsible for the phenotypic heterogeneity, the signaling dynamics heterogeneity. And we could show that 1 or 2 parameters are really important, the other 2 are not. That doesn't mean that they're not distributed, but their distribution is not important for the phenotypic difference, right?
Not that sensitive.
They're not sensitive, that's right. So in this case, what we did, our model has about 100 parameters.